talk will be in approximately equal parts in our work on crop nutritional quality improvement, primarily through breeding approaches in our group. I am a quantitative geneticist and plant breeder. And then I'll speak about some of our work on abiotic stress tolerance, namely to drought and high temperatures. Okay. So our lab was established at UCDavis just under 3 years ago. We are focused on phenotyping of crop nutritional quality, and that's part of why I thought to include this aspect of our work in scope today, given that you all, of course, are doing phenotyping for other traits. And I am hopeful that I can convince you that it may not be much more of a leap to look at nutritional traits as well, and you may have all of the other data sets needed to really dissect these. We do these genomic dissections as well. That is at the heart of our program, to identify genes and regions of the genome that could be helpful in improving crop nutritional quality. And then we work on actually implementing those breeding strategies as well to bring about the improved cultivars. We do look for those win-win scenarios. So our group has identified, in many contexts, nutritional traits are not yet valued along supply and demand streams. However, traits such as consumer preference, and that looks different in different crop types, of course, may already have value ascribed to them. So we end up looking for connections between some of these traits that are already prioritized and potentially nutritional traits so that we can deliver multi-value added products in the varieties that we're delivering. We do want to make sure that nutritional quality is making it all the way to the consumer. So we have a couple of projects that are looking at, is that nutrition actually bioavailable? Not just at time of harvest. Typically for genetic dissection, we would take those samples and put them into a -80 freezer, which is very helpful for a controlled study and for really high quality dissection. But of course, many of us would not tend to have a -80 freezer in our dwelling places. So we want to be able to take those samples through the post-harvest process and examine through processing and storage what do those nutritional profiles look like, which can be dependent on the crop genotype as well. So we see some really important patterns there as relates to all of genotype environment and management. And finally, we are working to integrate plant physiology and genomics in food crops, and it looks like you all have some projects that are likely parallel to this work stream of ours. We are doing this under stress, and we are adding in submodules for crop composition into these models, which have traditionally or classically focused on yield or biomass accumulation and partitioning. So I'll kind of hit each of these areas today with a little bit less focus on the delivered nutrition angle, but I'm glad to catch up on that if anyone is interested. Okay, so first we'll go through the nutritional portion rather quickly, and then we'll get to abiotic stress. So I thought to show you kind of how we do this dissection of identifying genes for nutritional traits. We end up using high performance liquid chromatography. This ended up being for vitamin E related compounds and provitamin A related compounds. There are other carotenoids that do not have provitamin A activity, but are also critical for eye health. So lutein and zeaxanthin are the two most abundant carotenoids in maize grain in this case, and have a critical role in the macular pigment of the eye, and are also important for age-related macular degeneration, which is a leading cause of blindness in adults as we reach our later years. We then have mapping populations. In maize, this is a neat population that allows us to leverage both ancestral or historical recombination in the form of diverse founders, and more recent recombination by producing recombinant inbred lines that have smaller sections of the genome kind of split up through subsequent crosses. We then also have expression data, so gene expression throughout the genome. We have RNA sequencing data at 6 developing stages through kernel development from 12 to 36 days after pollination. And that's really helpful as we look at the accumulation of these metabolites. We can also look correspondingly at the expression levels of genes that may or may not be involved. And then finally, we have high density genetic variants. These could be single nucleotide polymorphisms. We also have other genomic elements such as insertion deletions. We're starting to look at structural variants, which can be larger sections of the genome that can vary across individuals. But together, this gave us a really strong approach to be able to dissect several of the genes underlying both of these trait sets of interest to us. Again, vitamin E and provitamin A and their related compound classes. So we ended up for vitamin E, we were able to identify 14 genes, which do explain a large proportion of the variants for each of our traits of interest, which are indicated in the black font below here. Alpha tocopherol is the compound with the highest vitamin E activity. There are 2 other tocochromonols with non-negligible vitamin E activity. But effectively for any trait, we can take a look at the genes that could be contributing. Now we know that effect sizes change somewhat across populations, but we've done enough studies that we've really been able to identify and characterize the major players. For each of these traits of interest, or for a given gene, we can take a look at what set of effects it might exhibit. This includes looking at pleiotropy, where the same gene can affect multiple traits. And that's a particularly interesting set of questions in the context of a biochemical pathway, given all the dynamics that can go on. So I'll show an example of that in a bit here. But effectively, we may think, oh, well, we'll just identify biosynthetic pathway genes. So why bother doing the study to identify the genes? Turns out in this case, we had 8 of the genes that were in fact in the core vitamin E production pathway. Some of those, however, were in the precursor pathway, and some of them were related to chlorophyll biosynthesis, which was entirely unexpected. Maize grain is not photosynthetic or green, obviously. So we ended up really having some surprises there and kind of identified a novel biosynthetic cycle that's providing the tail of chlorophyll to produce vitamin E. So that was pretty interesting. And in terms of thinking about agronomic and nutritional links, that was a strange one, although in the grain tissue, it may be less interesting. And finally, we did have 6 genes with novel functions involved in, say, the storage and transport of these types of metabolites. So effectively, in either case, we end up having 2 to 3 major effect genes per trait, and that gives us a really clear way forward in breeding. So there are 2 major classes of these vitamin E related compounds, and for one of them, we hit those 2 homologs that are involved in chlorophyll biosynthesis. Again, this was entirely unexpected, but we did end up finding there were trace levels of chlorophylls in maize grain, specifically in the embryo, which again was not known previously, but it does look like these levels could correspond in a novel biosynthetic cycle sense with some of the vitamin E levels we're seeing. And finally, this other class, which are unsaturated compounds and largely in the endosperm of maize, we end up seeing somewhat more of an intuitive situation where we have a gene involved in the head group synthesis, a gene involved in the tail group synthesis for that same compound, and then a gene that helps to condense those 2. So whether or not we get surprised, we do have a clear way forward, which is, I would say this example contrasts with many other trait sets in plants. For nutritional traits, we may have a few major effect genes. We could call these oligogenic traits. And it's a very different genetic architecture that, again, really gives us major targets in breeding. Now, I would say when we go to traits such as drought tolerance, there have been limited examples in the literature where we see a 1 or 2 major genes being identified. But these nutritional traits really have lent themselves to this dissection and identification of major targets, whereas we otherwise need to use methods that integrate more signal across the genome. Similarly, for carotenoids, some of which have provitamin A activity, we identified 11 genes in this case, again, explaining the majority of the variation. And in this case, they were all biosynthetic pathway genes. However, 2 of these are really govern the production of what we call isoprenoids, or a large number of compounds that are stemming from this pathway. These 2 genes were also identified for the vitamin E compounds. So we see some interesting kind of interplay farther upstream for some of these classes of metabolites. And then we also, kind of leading into the pathway, we can look at what the different rate limiting steps have been classified. And some of that takes metabolic flux analysis, which is a really parallel method that we won't talk about today. And then we also, of course, need to consider degradation. So once we have that nutrition accumulated, we need to make sure that it's going to stay. So that's another strategy that we have at our disposal as needed, is to try to maintain the levels in the grain once we've increased them as far as we can. I will note as well, in case any of you enjoy orange maize, we do end up having, we did dissect in tandem maize kernel color, which ends up being a consumer preference trait, particularly in certain target regions for provitamin A enhanced maize. And we did find that kernel color, this is just one figure from that paper. We did a pretty neat random forest analysis too to see if we can predict kernel color from all these compounds, and reasonably well we can. But kernel color ends up clustering more closely with the beta branch of the pathway, which I showed on this previous slide, rather than the alpha branch. And it turns out that related to the double bond structure of these compounds, this makes good sense, but also suggests, based on what we found about the genetics, we might be able to improve kernel color alongside these beta branch nutritional compounds, which would be immensely helpful for, again, improving both trait sets at once. And this is work by Mary Frances Laporte in our group, who's a PhD candidate. She's sponsored by a fellowship through the Department of Energy, a computational science grad fellowship, and has been doing fantastic work. So just to give an interesting example to think about, we have 3 major genes that we identified for kernel color. And these graphs are depicting pleiotropy, which, as I described, would be when one gene is affecting multiple traits. So in this case, effectively, the blue is indicating positive pleiotropy, where if we have a positive effect on this compound, we end up having a positive effect on this trait as well. So we see for both this PSY1 gene and DXS2, both of these are upstream of the pathway, or this is the first in committed step, and this one's farther upstream, first in committed of the previous pathway. We end up seeing positive effects for many of the traits involved in this case. So not really incurring trade-offs with regards to which compound you would be increasing, because they would all be increasing. Whereas we do have this LIKE, which is the branch point enzyme, which determines whether substrate is going into the alpha beta branch. And accordingly, we do see if you have more of beta carotene as mediated by this gene, you end up having less of each of the alpha branch compounds. So in this case, you really do have to make a choice effectively with the allele or version of the gene that you're selecting upon. You may be effectively sending substrate into one or the other branch. So this suggests that we could be using some of these upstream genes to drive flux into the pathway, and perhaps not selecting upon some of these genes that incur trade-offs, except as is needed for nutritional benefit. So we can kind of prioritize nutrition when we have to make a choice, and otherwise can drive flux via methods that don't incur that trade-off. We are also working, we have a partnership with the International Maize and Wheat Improvement Center in Zimbabwe, and with our colleagues at the same institute in Mexico as well, to ensure that the hybrids being developed are performing well under stress. In this case, it's combined drought and high temperatures. We're also testing genomic prediction models. These use genome-wide markers, so looking at profiles across the genome to try to predict nutritional properties and agronomic properties for use in selection. This does eventually reduce the need for phenotyping. However, you need to continue phenotyping to keep updating your model to make sure it remains accurate over the generations. But these methods have looked really promising so far, and they can effectively be used at the seedling stage as well, because you can take a leaf punch, as some of you might be doing, and identify the genetic profile from there. Okay, we'll jump into the abiotic stress portion now. This is to change gears entirely, but I did, before coming to UCDavis, work at Corteva AgriScience as part of the predictive agriculture domain, and we ended up working on this method called crop growth modeling whole genome prediction. This is described in a somewhat recent publication as a GLM version of whole genome prediction, in which the CGM acts as the link function. So what we've effectively done here is we've placed a crop growth model in the middle of this prediction equation, where effectively for classical whole genome prediction, we might be looking at yield values and fitting marker effects for yield itself. In this case, what we could do is take observed yield values and run them backwards through this crop growth model, estimate certain physiological traits at the level of each individual cultivar or genotype, and then get those regularized to marker effects. So we end up having marker effects for physiological traits in this case. Here are a couple of examples, certain of parameters limited to transpiration in the case of that last one, and here's radiation use efficiency. So notably, now that we have those to marker effects, we could then estimate physiological trait values or breeding values for those, effectively for tested or untested genotypes. For any individual for which we have that genotypic information, we can pair the marker effects with the genotypic information, and we're leveraging relatedness of the individuals in that case. And also, because the crop growth model provides a mechanism by which to integrate environment and management information, we end up being able to run this model for tested or untested environments, provided that the assumptions of the CGM are reasonably accurate. And then we can run this model forward with those tested or untested genotypes, tested or untested environments, and reach a predicted yield value for any given set of these G and E and M, though we've done a little bit less on that so far. So there's a neat progression of papers where Frank Kernel ended up introducing this method, and then it was being applied across different types of breeding populations. And we decided to then apply this method across, effectively across a large breeding population. So we had what we call a half diallele, and these ended up resulting in a large number of maize families that we could look at, and we could look at the predictive abilities of this crop growth model, whole genome prediction, and classical WGP. So it turned out in this case, this is a little bit to look at, but I'll summarize it on the next slide too. So if we look only at the inclusion of well-watered data, which is characteristic of the Corn Belt here in the US, where most of the corn is produced here in the US, we end up seeing that the crop growth model, whole genome prediction, and the WGP end up performing about at parity with each other in each of the 4 quadrants of prediction. So for tested genotypes and tested environments, here we are for CGM WGP, the corresponding quadrant is here for WGP. So if we compare 0.79 to 0.74, 0.61 to 0.62, etc., we end up seeing these methods are performing roughly equivalently. Now, when we introduce water deficit data, now this would be for flowering stress, the timing of this drought stress falling at flowering, which is a critical stage in maize development, as many of you may have worked on. We end up seeing that the predictive ability of CGM WGP remains about steady. So comparing 0.79 to 0.8, 0.61 to 0.58, etc., whereas the predictive ability of classical WGP did drop substantially, so from 0.74 to 0.49, 0.62 to 0.42, etc. So this suggests that CGM WGP is able to maintain its accuracy when starting to include data from managed stress environments. And then I'll show a little bit more later, suggesting that it can actually help to integrate those signals as well in an informative way. The other key finding from this one, I'll just go to the next slide. Effectively, we don't see a penalty for including the crop growth model in the middle of that function that I showed. So even in only well-watered environments, it does not hurt to have that CGM in the model, was what we found in this specific data set, suggesting that the method may be okay to be using even in the relative absence of stress or the relative absence of extensive genotype by environment by management interactions. Another point that I found really interesting was that this method ended up producing stable estimates for physiological traits, and it could do so when you're testing with both well-watered and flowering stress data, and it can estimate multiple traits simultaneously, including for both yield potential and stress tolerance. So this ended up being helpful that the model really was producing stable estimates, and it was not kind of just trying to fit the scenario, which of course we have to worry about what we're fitting as we conduct this modeling. Well, we saw really promising signals there from testing a large number of scenarios. And I'll mention that, breeders, in addition to using across family predictive abilities, we end up using within family quite a bit as well. We did see similar patterns with the within family as we did with across family. So just in case anyone was wondering. So I saw you all have some modeling approaches, including 3D and even perhaps 4D. So I thought to show this example, but we do have effectively the way that this could work. Here's a crop growth model schematic that would operate on a daily timescale. And then we start kind of with vegetative submodules and then end up going into reproductive phase, eventually ending up with our yield and harvest index. So for one of these certain sub-processes, say we want to look at radiation use efficiency, we can look at how that is calculated within the crop growth model. These are mechanistic models in this particular use case. We're now testing other model types as well. But in this, we can then pull out one of the given parameters that say is known to vary by genotype. We can make sure that we have informative priors so that we know reasonable biological ranges for the material being studied. And then we get those to marker effects, as I mentioned previously. And then, of course, once we have that parameter, that can affect how the biomass accumulates and is partitioned and how that plant grows and develops relatedly. Another opportunity I'm really excited about with this method, which we explored in this paper, which is in Plant Physiology, if you'd like to check that out. We did look at 4 example crosses. So each of these NSS is a parent. And it turns out, so we're looking here at principal component biplots. And in this case, we can really look at how a given cross shows up in germplasm space. So it turns out that if we cross 2 elite parents that do not have that drought tolerant phenotype, in this case, we ended up seeing above average for the population values for senescence. When this is a trait that relates to whether that plant will stay green throughout the season and continue to photosynthesize later into the season. So it turns out this high values for senescence correspond to not having that stay green trait. So effectively, this cross is not going to end up having the stay green trait that could be helpful in certain drought conditions. Whereas we can look, as we start to cross with a drought tolerant parent, we can look to see how that germplasm effectively has moved around in trait space, and including as relates to those yield vectors. So looking at how yield could fall out in a well-watered, water deficit, et cetera, situation. So this can be helpful as a breeder. This could be helpful to me in looking at which crosses might really be exploring unique portions of the physiological trait space, which ones might be producing kind of largely overlapping clouds of progeny, which again, overlap may not be a bad thing if you know exactly where in that space you would like to be. But this does help us to look at kind of how these crosses, whether their strengths being complemented or vulnerabilities that might be exacerbated by making a certain cross based on kind of the trait values that are present. Okay. Yeah, and I'll mention, I guess we ended up having an optimal model in this case that had 4 traits involved, and we did kind of a forward selection procedure for deciding which traits would be in the model, just to anticipate, in case that's a question. I saw you all are also extracting parameters for certain modeling efforts, but we ended up having a couple that could be relevant under drought stress and a couple that are more focused on yield potential. We also have a project in sorghum that's combining kind of the two sections of this talk. We're doing more studies of this sort where we look at both productivity and nutritional quality in tandem, again, with the goal of having that multi-value added product development. So we are looking in this case at pre-flowering drought. This is when grain number is being determined, and this is in grain sorghum now that we're doing this. So we want to look at the effect of that drought stress on grain number, and then look as well at impacts on grain quality. We are developing these genomics assisted selection methods. We are also testing out this crop growth modeling and integration with genomics. And this is one of the projects where we're starting to build in those compositional traits and see, okay, well, we have a little bit of a lead within the model for, say, grain nitrogen, because it's been thought to be connected to grain growth rate in the past. But can we now actually build out that submodule and get more of the grain compositional parameters really integrated? So talking about things like protein, fat, starch, perhaps total phenolics, which could be related to the grain color, which again is more important in some crops than others. But that is what we're looking at there. And that work is also in partnership with Dr. Julia Sabia, who is at the University of KwaZulu-Natal. And they're also testing an abiotic stress and nutritional quality angle. So we've really enjoyed that collaboration with a South African breeding program. So we ended up studying, effectively, we grew sorghum field trials 3 hours south of Davis. And this ends up being a really important site for the University of California that is operated through Ag and Natural Resources here. And we end up growing what this is an association panel that's more leveraging that ancestral or historical recombination, but allows us to dissect various traits. We had pretty good repeatability, so we were using an augmented design here, and I saw you all are also working on new field mappings, but we had an augmented design where we have repeated checks in each block and then can correct for that spatial variation. So we have pretty good repeatability within treatments across replicates for some of the agronomic traits of most interest. Nutritional assays are still underway, and that's using near-infrared spectroscopy in that case. And finally, I'll just describe briefly, we do have some integration of AI-enabled prediction and of 3D modeling frameworks. And this has primarily been a collaboration with Mason Earls and with Brian Bailey here at UCDavis. And here are some figures from Brian that are shared with permission, where Brian and team have a 3D biophysical modeling framework. And actually, I would nominate both of them as speakers. And if you all have not already spoken with them, they give excellent talks and are really doing fantastic work. But Brian and team have developed 3D biophysical models, and Brian has developed a software platform and really great documentation to accompany it. And they've looked at different systems. They've modeled here a Stanford dragon, which I suppose is a classical object to model, and are also looking at some orchard crops. And are really looking at, again, some of the sub processes that I discussed, but they can really get the biophysics in there. And Brian has an engineering background and has been bringing his expertise in that realm. I will mention we are testing some of these methods out in common bean and other crops in which we see a certain type of occlusion. So that's something that Mason and Brian have been dealing with in other crops. But in the case of common bean, this is the crown of a common bean plant where a large proportion of the beans may end up accumulating and ends up being quite in the understory. So we are thinking about, for some of these nutritional security crops, these common beans are among the most direct beans for human consumption and a really important source of protein among other micronutrients. So we end up wanting to do some of this prediction for productivity and quality. And we have a large network of partnerships focused on doing so. And thankfully, a very strong team here. Sassoon Lowe is a cowpea breeding and genetics expert who has joined the project. Johnny is a PhD student, and he is funded by this AI Institute that Elias introduced. And Earl is one of the AI or AIFS career exploration fellows who's so finishing undergraduate this year. All three of them have been doing absolutely stellar work and in this space of integrating AI-enabled sensing and prediction and 3D modeling into the molecular breeding process. So I think that's everything. Hopefully I can proceed. So I did present a fairly large number of projects today. This Vitamase project group was funded by NSF to complete the nutritional work that I discussed. We just got that paper out, the simultaneous carotenoid kernel color work. CGM WGP team, again, this is a team from Corteva that completed that work, and many thanks to them for supporting that effort and getting it published. Summit Zimbabwe, NIFA Sorghum, that APHIS project team. We do have support from APHIS for a project I didn't go into much detail today, but again, I'm glad to talk about more. And funds from NSF USDA and Cornell and UCDavis as well, and USAID. And then we had a large number of, again, of projects represented, but this is where we are, and I'm glad to discuss and looking forward to it.